In this video, I want to introduce you to the mechanisms of cellular motility used by cells to move through their environment. We want to quickly cover two different mechanisms, actin-based, which uses actin and myosin motors for cells to crawl across their surface, and microtubule-based, which uses microtubules and dynein and kinesin motors for beating cilia and flagella. And we are going to be looking at Chlamydomonas reinhardti, a single-celled algae that has two flagella this week, to actually look at some of that movement or motility. First kind of cytoskeleton filament that's involved in movement is the actin filaments. Actin filaments are the smallest filaments that use actin dimers to build this longer filament. And there's going to be two ends. There's going to be a minus end and a plus end, so it has polarity, and that's important so the motors know which directions to walk on these filaments. If we look closer at this, what we realize is that at the plus end we add on our actin monomers, and generally at the minus end we lose them. So these treadmill, meaning that we are adding onto one end and losing from the other end over time. But this makes it also a very regulatable system that we can control. This system is often used for moving a cell by having growth of actin filaments at one side of the cell and shrinking of those actin filaments at the other. And so if this is the leading edge of the cell, meaning the cell is moving in this direction up, what the cell does is it starts to polymerize new filaments at the plus end here right by the membrane. And those new filaments that are being polymerized push on the membrane, so push the membrane upwards. Back here at the trailing edge, these filaments are breaking down, and then those actin monomers can be brought forward and recycled and used again. So what this sets up is amoeboid movement or crawling across the surface. So this is the cell that we're moving in this direction forward, and we start polymerizing new actin polymerization at the tip. The movement of the unpolymerized actin then is brought forward so we can continue to push forward. We make focal contacts here on the surface to hold the cell down, and then myosin motors pull up the back of the cell, and so the whole cell will crawl forward. Okay, so that's actin-based movement.